Hello, this is the next set of video lectures, and the topic is non-Mendelian genetics. As we mentioned in last class, with basic genetics, there are simple crosses that can be done when a trait only has two options. For example, if you're talking about the presence of freckles or the absence of freckles, those are the only two options. However, most traits have more than two options in which to pass on to the next generation. Those would be part of non-Mendelian genetics because it's not a simple dominant and recessive relationship, since there are multiple combinations that can occur. So since there are multiple combinations, there ends up being more complex patterns of inheritance. So these more complex patterns of inheritance do not fall under Mendelian inheritance, just controlled by dominant and recessive alleles. We still have some of the principles that Mendel shared, segregation and independent assortment, applying to these more complex patterns, but basically there are a couple of different ways that these other patterns can arise. First of all, alleles may or may not be completely dominant or recessive. At times, a gene may have more than two alleles, although each individual will only carry two. And then sometimes a gene may even produce more than one physical appearance or phenotype. The first non-Mendelian pattern I'd like to discuss is something called incomplete dominance. As its name suggests, there is not a completely dominant allele and a completely recessive allele. In Mendelian genetics, complete dominance has heterozygous and homozygous dominant individuals having the same phenotype or physical appearance. With incomplete dominance, the phenotype of the heterozygote is an intermediate between those of the homozygous recessive and homozygous dominant individuals. So this is the case where the heterozygote actually shows a blending phenotype. This happens because one allele is not completely dominant over the other. In the picture on the right, we have a red flower crossed with a white flower producing pink offspring. Let's look at an example more closely. If you cross a white flower with a red flower that exhibit incomplete dominance, the first generation will all be pink, phenotypically, and they genotypically will be heterozygotes. However, if you cross two of those heterozygotes, the parent phenotypes, red and white, will appear in the F2 generation. So a crossing of two heterozygotes will result in a 1 to 2 to 1 phenotypic ratio. 1 red, 2 pink, 1 white. For incomplete dominance genetic problems, we can still use the Punnett square to solve these problems. The trick is to recognize when you're dealing with a question involving incomplete dominance. There are two steps. First thing, notice that the offspring is showing a third phenotype. The parents each have one, and the offspring may be different from the parents. Secondly, notice that the trait in the offspring is a blend or mixture of the parental traits. So let's now go ahead and do some incomplete dominance questions. So this is where I will have you stop so that you can work through these, because I would like you to know how to do incomplete dominance questions using a Punnett square. Number 1. A cross between a black bird and a white bird produces offspring that are gray. The color of birds is determined by just two alleles. What are the genotypes of the parent birds in the original cross? For incomplete dominance questions, I use two different letters and recognize that when they're combined, the third or heterozygote is going to be a different phenotype. So instead of using a capital B for black and a lowercase b for white, I use a B for black and a W for white. I do not use any lowercase letters, and I recognize that when the B and the W both exist as the heterozygote, that's gray. So in this case, for part A, I use two capital B's to denote the black bird, two capital W's to denote the white bird, and a capital B, capital W to denote the gray bird. So for part B, what is or are the genotypes of the offspring? Capital B, capital W. What would the phenotypic ratio of offspring produced by 2 gray birds be? This would be 1 black, 2 gray, and 1 white. And sorry, but there's that Punnett square that shows you 2 gray birds, the capital B, capital W, that the 2 capital B's show up once again, representing the black bird, and the 2 capital W's show up once again, representing the white bird. A second one. The color of fruit for plant X is determined by 2 alleles. When 2 plants with orange fruits are crossed, the following phenotypic ratios are present in the offspring. 25% red, 50% orange, 25% yellow. The question, what are the genotypes of the parent orange-fruited plants? Well, this is where you recognize that orange is actually a blend of red and yellow. So using the same way that we did question number 1, red would have 2 capital R's, yellow would have 2 capital Y's, so orange would have or use a capital R and a capital Y. The next pattern of inheritance that I'd like to discuss is called codominance. This is often confused with incomplete dominance. In codominance, however, 2 dominant alleles affect the phenotype in separate yet distinguishable ways because both will be expressed in the animal or offspring or whatever you're talking about for the cross. Co, remember, means together, so codominance means there are 2 alleles that are equally dominant. The example I have in the picture at the right is what's called a roan cow. A roan cow has a red coat with white blotches. So the cow is not red in its coloration and it's not white in its coloration. And if this were incomplete dominance, that means a shade in between would appear. And of course, we don't see any pink cows or anything where there's a mixture of those two. But in the cow at the right, both are showing up. So you see parts where there's the red coat and parts where there's the white coat. So this causes codominance to occur. Again, this is not a blending. Incomplete dominance was a blending. Codominance is where both are fully expressed. The gist is pretty much the same though. There is a third phenotype, but it's not the usual dominant or recessive one, and it's also not the blending. In codominance, the recessive and dominant traits appear together in the phenotype of hybrid organisms. So red and white would create red and white spotted. Sometimes there might be an additional allele, but we'll talk about that in a few moments. Let's practice using a Punnett square, questions using and involving codominance. Some texts use letters and superscripts when dealing with codominance. Others use different letters, as we just have with incomplete dominance. So I'll use the second method for our example. A capital R will be the allele for red flowers. A capital W will be the allele for white flowers. So when you cross those two, instead of getting pink flowers, you get red and white spotted flowers. The symbols you choose don't really matter as long as you remember this is a codominant genetic inheritance problem, because this is the same letteration that we used for incomplete dominance. For codominance, when you cross 2 capital R's and 2 capital W's, 100% of the offspring are capital R, capital W, which is red and white spotted. And if we took that to the next generation, we would see that red and white spotted flowers crossed with each other would produce both the pure red and the pure white along with red and white spotted in a 1:2:1 ratio. The 2 in the 1:2:1 ratio would be the red and white spotted. So let's now take a look at some practice problems. Number 1. Predict the phenotypic ratios of offspring when a homozygous white cow is crossed with a roan bull. And again, roan is a vocabulary word that I'd like you to know, meaning that roan is red and white appearing together. Well, here we have our answer. The white would be 2 capital W's, the red would be 2 capital R's, and the roan would be a capital R and a capital W. So when you do the phenotypic ratio, the two capital W's mixed with a capital R and a capital W will give you half white and half roan. Number 2, a cross between a black cat and a tan cat produces what's called a tabby color, black and tan fur occurring together. Letter A, what pattern of inheritance does this illustrate and why? The answer being codominance, of course not only because that's what we're talking about, but because both alleles are appearing together. And then also B, what percent of kittens would have tan fur if a tabby cat, which is the heterozygote, is crossed with a black cat? So taking you through that, tan would be 2 capital T's, black would be 2 capital B's, meaning the tabby color would be capital T, capital B. So when you do that cross, capital T, capital B, combined with 2 capital B's, half of the offspring would have a tabby pattern and half of the offspring would be black, so that means 0% of the kittens would have tan fur. Those are just 2 Punnett squares to illustrate the crosses that were done in those problems. The next idea involved in non-Mendelian genetics are multiple alleles. This is the one I said we'll get to in a moment. Multiple alleles is exactly as it sounds. It is very common for more than 2 alleles to exist for a trait in a population. This is one of the things I mentioned in the beginning of this lecture, saying that non-Mendelian often deals with the types of inheritance patterns where there's more than just 2 options. Multiple alleles exists when you have different forms but more than just 2. So this is not the case where freckles are either present or absent. This actually applies to a lot of human characteristics since we see great variation in the population and multiple alleles are said to be involved. A diploid individual, however, can only possess 2 alleles. So if there are 3 or 4 alleles for an individual trait, any individual themselves can only possess 2 alleles. And then looking at the inheritance pattern would determine what your physical appearance would be based on your genotype that you have. In this picture, it's just showing you the multiple alleles that exist for different rabbits. In here, you'll see that there are superscripts used because there's not more than 2 versions of a letter. So using the superscript will give us more alleles to use in a regular Punnett square. So there's the capital C, the lowercase c, and then there's the C with the superscript CH, and then there's also C with the superscript H. That tells us that there are 4 different alleles here, multiple alleles, and the combinations result in 5 different phenotypes, ranging from dark gray to albino. Well, the number of alleles is not limited to 4 because there are instances in which there are more than 100 alleles existing for a single trait. The most common example of multiple alleles involves our blood type, human blood typing. Multiple alleles do govern blood typing as well as one of the previous inheritance patterns, codominance. Human blood types are actually determined by the presence or absence of certain molecules on the surface of red blood cells that we call antigens. In the chart at the right, it tells you the phenotype, which would be our blood type, A, B, or O. The genotype is represented in the far left, and in the middle column, the appearance shows the different antigens that are present. We write the phenotypes just by what the alleles are. So either A, B, or O. The genotype is written using a capital I or a lowercase I. I'm not exactly sure why the I was chosen, but the capital I would either carry the superscript A or B since those are both dominant over the recessive allele, which is O, denoted by the lowercase I. So the capital I with a superscript A is the allele that produces antigen A. Antigen A, on the diagram at the right, has a triangle appearance attached to the surface of the red blood cell. Capital I with a superscript capital B is the allele that produces antigen B, which is a sphere attached to the surface of the red blood cell to the right. The lowercase I, which represents the O allele, produces no antigens, which explains why there's nothing attached to the cell surface of the red blood cell in the type O individual, whereas the type AB individual produces both the triangle and spherical antigen markers on the surface of the red blood cell. This also explains why there are universal donors and universal recipients when it comes to blood types. Antigens are substances that would elicit a response from your immune system. Therefore, if you are the type O individual, your body is used to not having anything on the red blood cells. If you are given blood from any other blood type, be it A, B, or AB, your body will actually attack those blood cells. So type O individuals can only receive blood from other type O individuals. However, since there aren't any antigens on the type O red blood cell, they are able to donate to any of the other blood types, A, B, AB, or O, which is why type O is considered the universal donor. Type AB is actually the universal recipient. Because they have both types of antigens on their surface, they're able to receive and use either type A blood, type B blood, or even type O blood because there are no antigens present, as well as the obvious type AB blood. So type AB individuals are said to be universal recipients. It's important to know blood type because incompatible blood types would clump together, causing death. It's also important when looking at disputed parentage, which you may have seen or come across in some daytime television shows that are like Jerry Springer. If a child has type AB blood and its mother has type A blood, a man with type O could not be the father. And the question I ask is why? A man with type O blood, in order to phenotypically show type O, he would have to possess two recessive lowercase i's, which means two recessive O alleles. If the child has two dominant alleles, A and B, which, by the way, are codominant, both dominant to the recessive O, that means the A in the child's blood type came from the mother, and the B in the child's blood type had to come from the father. If a man has type O blood, he is definitely not carrying a type B allele.